My name is Savan Shitalanil. I am from SYBSC and today we studied how to make the DNA isolation buffer solution. For this we took five types of the items. That names are Tris HCl having pH 8.0, EDTA.Na2 which having pH 8.0, NaCl, CTAB which is used for plants and fungal, and or SDS which is used for animals, and the last one is double distilled sterile water. So first we make the stock concentrated solutions of all the items. So for the trace HCl we have 1 molar stock concentration of it, and for EDTA which is 0.5 molar, for EDTA which is 0.5 molar, for NaCl which is 5 molar, CTAB or SDS which having the stock concentration of 20%. So for the working concentration we have 100 millimolar of trice HCl, we have 50 millimolar of EDTA.Na2, and for NaCl we have 1.4 molar of the working concentration of it. For the CTAB or SDS, we have 2% or 1% of its working concentration. We have to make the 100 ml of total concentration. Out of the 100 ml of total concentration, rice HCl is used 10 ml, the EDTA is used 10 ml, NaCl is used 28 ml, and CTAB or SDS is required 10 ml, and the last one is double distilled sterile water which is required of 42 ml out of the 100 ml concentrations. Then we have to take 5 bottles of 100 ml of each. In the first bottle we took 1 molar trice HCl having pH 8.0, and we mention here the date on which we made this, that is 18/4/2023, which is made by V.K. In the second bottle we took 0.5 molar EDTA having pH 8.0. We made this on 18/4/2023, which is made by the V.K. The dates and the name are mentioned because that is very important. We have to know about the expiry dates and manufacturing dates, so that is required, and we mention it on the bottle. In the third bottle we took 5 molar NaCl, and which is made on 18/4, made by V.K. In the fourth bottle we took 20% of SDS. We mentioned here the date 18/4, which is made on this date, which is made by the V.K. In the last bottle we took double distilled sterile water, in the 100 ml of bottle, and we mentioned here the date on which we made this 18/4/2023, which is made by the V.K. So from these all bottles we have to make the DNA isolation buffer solution. So we took 10 ml from trice HCl, 10 ml from EDTA, 5 molar NaCl which is required 28 ml, and from the 20% SDS we took 10 ml, and the last we from the double distilled sterile water we took 42 ml, and at last we made all by adding all these solutions we made the 2% of CTAB DNA isolation buffer solution in the 100 ml of water on the 18/4/2023, which is made by V.K. And this DNA isolation buffer solution is used to extract the DNA from the working samples.